So in section 3, we're going to start talking about Gregor Mendel and the work that he put into I basically becoming the father of genetics. So Mendel laid the groundwork for genetics. He was an Austrian monk that he didn't go about saying, like, I'm going to figure out how genetics works. He was in the monastery. They had other jobs that they're supposed to do, and one of the jobs that he chose to do was work in the garden of the monastery, and he wanted the plants to be a particular way. And so he was very meticulous about his notes and information that he wrote down on how he ended up getting the plants to be the way that he wanted. And he was basically figuring out genetics at the time. So one of the things that he did is that we know that traits are distinguishing characteristics that are inherited from a parent. So genetics is the study of biological inheritance patterns and variation. Mendel showed that traits are inherited as discrete units. So many in Mendel's day thought that traits were blended. So you would always see a mix from the two parents that were involved. So it was something unique that he ended up being able to figure out and show that it was not that case. So Mendel's data ended up revealing patterns of inheritance. So that those units of traits that we were talking about, we refer to those as genes now. So Mendel made 3 key decisions in his experiments. He used purebred plants. The plant that he used was a pea plant, and he ended up with the pea plants, chose them because they were didn't take up much room. They were quick growing, something that they could eat. But they are a good use in the science experiment because of some of the things that it doesn't take long for them to mature, things like that. So another thing is he had control over breeding with it. So he could prevent them from breeding if he did not want them to. And the other key thing was he ended up doing observations on 7 key traits that were either or traits. You had this or you had that. So tall or short, for example. Mendel used pollen to fertilize selected pea plants. The P generation, which the P stands for parental, crossed to produce the F1 generation. The F1 generation, F stands for filial, which means son. So the P generation gives the F1 generation. Mendel ended up interrupting the self-pollination process by removing the male flower parts. So that's what you see down here is him basically cutting off the stamen of the flowers and then putting them in jars, and then he would make sure he knew which jar was which, and he would be able to take the pollen on a paintbrush and go pollinate the flower that he wanted to have it reproduce with. So the Mendel allowed for the resulting plants to then self-pollinate. So among the F1 generation, all the plants in his experiment, he bred white and purple flowers, and all of the plants ended up having purple flowers. So the F1 plants ended up all being what's known as heterozygous. So that is a mix of alleles. Among the F2 generation, so he took 2 plants from the F1 generation and had them self-pollinate or cross with another one from that same generation. And all the F2 plants, some of the plants had purple flowers, some of them had white flowers. So the trait that he saw that had disappeared reappeared in the F2 generation. And that's what we see down at the bottom of the screen there. So crossed two true breeding plants, and the true breeding plants produced a hybrid, which is in that F1 generation, and then had those self-pollinate or those two cross, and you ended up with some that were purple and some that were white. Mendel observed patterns in the first and second generations of his crosses. So you can see if he was looking at the ratios and categorizing them, always was right around a 3-to-1 ratio is what he was seeing of dominant to recessive. Mendel drew 3 important conclusions from his work: that traits are inherited as discrete units, which are called genes. Organisms inherit 2 copies of each gene, one from each parent. Those copies are called alleles, that we'll talk about in a little bit later. The 2 copies segregate during the gamete formation. So those alleles that I mentioned, the different versions of that gene, are going to separate from each other when a sex cell is made. So they're not going to both get passed on together. The last 2 conclusions are that are called the law of segregation. And we'll talk about the law of segregation in the next couple of sections here. But the law of segregation basically says that you're going to inherit a chromosome 1 from your mom or dad, or like a mixture of that. You're not going to pass on 2 chromosome 1s to your offspring. They're going to separate independently from one another, and you're going to not have them affect one another as well. So that is the end of the notes for section 3.